All right, well, hello, good morning. Welcome back to another virtual visit here at the Alaska SeaLife Center. As always, I'm Alex, and today I'm joined by another member of our education team, Taylor.
Hi.
You may have seen Taylor before you've done a couple other programs, and today we're going to be catching up on what Taylor was doing last week while we were getting all set up for that virtual visit, because we're actually talking about something that happens on Wednesdays, and that's, of course, when we do our virtual visits, is every Wednesday at 11 a.m. Alaska time. And we want to thank our sponsors for these virtual visits. This year, Royal Caribbean Group has sponsored our whole season of virtual visits, so we can bring those to you live and free. It's nice to be able to just expand our audience with these and get them out there to whoever is interested in knowing kind of what goes on behind the scenes of the Alaska SeaLife Center or what's happening out on the floor any given time. Now, speaking about what's going on, let's take a look at our sunrise today. As always, this is from about 8:30 or so in the morning until about 10:30. Now, the sun has been getting up earlier and earlier in our recent sunrises. However, you don't really see it in today's sunrise, and that's because we're kind of cloudy weather today. In fact, we've almost got a little bit of fog. It looks like that comes in, but it's really just these low clouds kind of settling down into Resurrection Bay. And someday we're going to have to do an episode of Resurrection Bay. And just the cool things about it, one of the big things is it is a glacial fjord, which means that it is very steep-walled. It's carved by a glacier, long time ago. It's actually shaped kind of like a bathtub, although the sides are very, very steep. The bottom is very, very flat. And so clouds like to settle in here. We've had, smoke from forest fires elsewhere in the state, or even forest fires out across the Pacific. We've had that smoke settle into Resurrection Bay, so kind of one of the problems here. And on a cloudy day like this, it's not uncommon for one of those clouds to kind of just sink on down, and so that's what we saw there.
I think cloudy days are one of my favorite days here in Seward. They're very cozy.
Yeah, they are cozy. They're not bad. Seward doesn't get that cold. If everyone's like, oh, they're in Alaska, it must be freezing. It's not that cold. It's right around freezing today. It's actually above freezing right now. We've got a little bit of rain. And then at night it'll dip down into freezing. We'll get a little bit of snow, but we don't get like the negative 40s that you hear about in Fairbanks and that sort of thing.
Thank goodness.
Now, for anyone who's tuned in for these virtual visits before, this is a place that we aren't normally hanging out in, but this is a place that we have been before, and this is what you were looking at while you were waiting for our program today. We were looking at our central lab. And so if you've tuned in for the behind-the-scenes visit, Taylor got to kind of chase me around as we came in and filmed all the areas in the center. And so you hung out here last week.
I did, and I had a great time. And it's really awesome that they're actually running water quality behind us as we speak.
Excellent. So that's what we're going to be talking about today, is that water quality. So before we jump on in, you had the chance to follow them around basically last week. You were back there. So if anyone sees any like commotion going on back there, or our mics are picking up some weird machinery noises, water quality is very important. It happens every Wednesday, and then it actually extends into Thursday, so you had 2 days of work.
Right, yes, because it has to go into an incubation period. So there's some cool machines that you'll see here in a second where then it goes through, it incubates, whether it's in a water bath or in just a normal incubator, and then come back the next morning pretty early. I think we were in here at like 7:45 the next morning reading water quality.
Pretty early. So you just want to jump on into the first day?
Yeah, let's go for it.
All right, let's see what you checked out for water quality.
Now, water quality is such a crucial test that we run at the center. Here you can see all of the bottles used for collection for each of our pools and habitats. Laura, one of our aviculturists here, is coming to grab the bottle for the aviary, and we got to tag along as she collected the water. These bottles have all been sterilized, so this is the point where she can take the cap off. There is a very, very specific motion whenever collecting the water. You can put it out as far as you can, break up the surface tension of the water, invert it where you broke up the water, bring it back up, and pour out just a tad so the vet staff can shake the bottles to prevent anything from sitting on the water when they run the test. Let's learn a bit more from our vet staff.
We run water quality every week here at the SeaLife Center to keep track of all of the pools. It is a mandate by USDA to run water quality samples on all of our marine mammals here at the center, but we also run it on our bird habitat because we want to keep everyone at a very, very high standard of health. For USDA, it's mandated that we are running a test called total coliforms, And then for our divers, because we have divers going into the habitats, we actually have a much more specific test for human safety called enterococci, or ENT, and we run that every week as well.
One of the first steps in starting water quality is measuring out 90 milliliters of sterile deionized water, or DI water, for each of the bottles. Now these bottles have been sterilized by an autoclave, which is why they have the piece of indicator tape on them that reads a black line when they have been properly sterilized. So an autoclave is a machine that is designed to get up to certain temperatures to sterilize all parts of equipment. It is very important that we have autoclaves in vet clinics to sterilize surgical tools, and also really important for water quality here because the bottles and everything being used should be as sterile as possible because we do not want any outside things interfering with what the pool would actually be giving us or reading. Now our vet staff is going to be conducting 2 water quality tests, as Hannah mentioned earlier. One for TC, or total coliforms, which coliforms represent a group of bacteria. So there are many, many different types of bacteria that fall underneath this coliform group, and they're primarily found in fecal matter, which is why we are testing these pools and making sure they are within the USDA health standards for our animals here at the center. We also run a test looking at ENT, or enterococci, which is the most accurate test. So this is why we use ENT here when regarding diver safety and making sure humans can enter the water safely as well.
We test ENT for all of the habitats that our divers are in. So we have stellar sea lion habitat, seal habitat, birds, and then we have our control. This is the packets that help, they're the reagent packets that test for ENT. So each one of these, put that one. And so the most important thing is we have to shake these bottles to mix them so that you're getting a representative sample, so that anything that's sitting on the top or the bottom gets mixed around. I'm taking 10 milliliters from each bottle and putting it into my sample bottle. 90 milliliters of sterile DI water plus 10 milliliters of the sample water so that it's a 10x dilution. We don't want to create any bubbles, so that's why I'm swirling it to dissolve the powder. This is stellar sea lion habitat, so Pilot's in here right now. So this is our Quantitrays. So what I'll do is I'll pour the water into here, and the water will sit in each of these wells. So then I just pour it in. And then you can see, you kind of just want to tap out the bubbles. Because you want all the wells, there's 51 wells in each of these trays, and you want all of them to be filled once we put them through the sealer. Each well represents a value, and it will give us an idea of the total number of coliforms, or the most probable number of coliforms, and I can show you later how we determine that. When we read the results, it'll be a little more clear. But so yeah, now we just want to seal it. So I lay it down, make it flat, and I run it through the sealer. And that's just going to even out and make sure that all of the wells have water in them. So yeah, I just want to make sure that all of the wells, it's okay if they have a little bit of bubbles, it just has to be mostly filled. And this top one also needs to have water in it as well. So that looks pretty good. So I'll just set that aside for now, and I'll do the other ones. so now that they're all ready to go, we're going to incubate them in a water bath. So this is set to 41 degrees Celsius. And so they incubate for 24 to 28 hours. So you just kind of set them in here. And so in the morning, we'll read them and we'll be able to see what our tanks are reading at for ENT.
All of these pools are cleaned regularly. I would say that water quality, yes, it's mandated by USDA, but it's a great checks and balances system for us to make sure how we're cleaning our pools is actually effective. We have been talking about maybe incorporating different types of cleaning solutions to cover different types of bacteria. I know that like COVID-19 really brought that up. Like I personally have been looking at different disinfectants because of that. So what we'll do is we'll move the animals out of the pools, like our ODLs and our back pools. Animals will be moved out, we'll drop that pool, we will rinse it really well, we will scrub it with a Dawn solution or just a cleaner, and then we can use a disinfectant such as a dilution of bleach. We can use a dilution of something called Excel, which is used in other like vet clinics and things like that. And we will also use chlorhexidine solution. For our habitats, it's really hard to actually drain the habitat. So we rely on divers to go in and siphon out waste and scrub the windows and things like that. So that's why we have to test ENT, because if it's not safe for the divers to go in there, then we have to really change things up.
During water quality testing, I also had the opportunity to ask Hannah what she does here at the SeaLife Center.
I am a vet assistant, so I help out in a lot of different areas. So obviously I help out in our lab, I do water quality. Our lab also will process blood samples, fecal samples, urine samples, so I help out with that. I also participate in collection of those samples and maybe restraint of animals during medical exams. And then I also help out a lot in our rehab department, which is probably like my favorite part of my job. So this is actually a different color than our ENT test, so you'll see when we pour it in, it's a lot lighter.
So what a cool process that I got to experience and sit in on for day one. I came in at about 8 in the morning and we ran through that, gosh, it was probably a couple of hours where I got to understand the first day.
And I was gonna say, I mean, that's just the first day.
Just the first day.
We haven't seen day 2 yet.
No.
So it's a lot of work.
Absolutely.
And they do this every Wednesday. As we mentioned, we are actually back in Central Lab. We're tucked in like a back corner. So you might have seen them kind of working back that way. You might have seen them coming back and forth. But we just wanted to tuck out of the way because they have to do this every single Wednesday, and we wanted to be in here for it, but it overlapped with our recording time.
Yeah, we were just trying to make sure that we were trying to be quiet over here in the corner, but as you know with the live program, it's always kind of difficult. Speaking of live programs, if you have any questions at all, please feel free to send it into our chat if you're live, or send it to our text message, which is in the description below. We'll be happy to answer those. Why don't we get a look at day 2?
Look at day 2, all right. And I do want to mention, of course, if you're not watching this live, please leave a comment. We can still answer those questions even after the fact.
Our water samples have been incubating for about 22 hours now, so we're going to take them out and read them. So this was our TC test. right, we'll start with our ODL ones here. So, as you can see with this tank here, there's nothing with ODL 5, there's nothing lit up. This is what we're looking for. When it's a yellow well, that means that it's positive. So if we look at our well count, this is how we read them. So for ODL 6 with that one well lit up, we would say the most probable number is 1. And then we multiply that by 10 for our dilution that we did. So it would be the most probable number is about 10 coliforms. So this is where we get into where I have to count them and make sure that they pass. We'll go to 15, so that's 17.8. So 178 is the most probable number for dilution. So what I'll do is I just keep track in this binder. Stellar seal habitat and seal habitat. The next thing we're gonna do with these is we're gonna use fluorescent light and see if any of these fluoresce. And that's indicative, it kind of just tells us about the certain bacteria that it could be. So this one's good. This one's good. So you can see that this well is fluorescing, and that's also one that I marked. So I'm gonna go ahead and just put an X through that.
So we use the IDEX Call Alert 18 method to do our water quality. We look for yellow wells. That means that there's a positive presence of coliforms in that sample. Up to a certain number of positive yellow wells in this method, we would have to say a pool passes or fails. What we also look for is the fluorescing when we turn off the lights and shine it with a UV light. That's another property of this method that we can just use to see if there is E. coli present. So we don't necessarily have to keep track of that per USDA regulations, but with this method, it's another data point that we can track. So we just like to document that. And it can tell us that there is presence of a bacteria of a coliform that we should be aware of.
The SeaLife Center houses many animals and has the great responsibility of ensuring that these animals here are safe and well taken care of. And one of the ways that we do this is by maintaining the health standards of the habitats in which they live. Although these water quality tests are done behind the scenes, they are an essential part of caring for the animals the public loves to see during their visit to the SeaLife Center.
All right, well, welcome back. That was day 2. So it went a little quicker, it seems like, but it is still a lot of work actually going through I think it was like blister packs. Like if you ever bought like action figures as a kid or something, they come in like the little plastic bubble. Like that's what it is in my head. But they kept calling them wells.
Yes.
And how, there's one per tank basically?
Yes, absolutely. There's one per pool or habitat, and there's 51 of them. There's 51 wells. So as you can see, we're looking to see which ones may be lit up either a yellow color or when it fluoresced underneath the UV light.
Yeah, okay. And actually, it looks like we did have a question, so maybe I can get into this. We said, they're doing this per like habitat, but they mentioned it's only mammals and only birds that they're really doing this with. So why don't we run water quality on aquariums?
Yeah, well, fish are not USDA regulated or mandated, and so our team here, our aquarium team, is super, super great at keeping up with all of our filters and making sure. So do you have more perspective on that?
There are different tests. It's not this test. It's not this test that they run here, but frequently you'll actually see our aquarium staff kind of going around. If you've ever seen them going around and they've got like the long rubber cable and they like kind of drop it in the bird habitat, which has fish, they drop it all the way down there, or they'll do it in our salmon habitat. Folks see this sometimes when they're visiting the center. They're actually looking at like how much dissolved oxygen there is, and they're watching the temperature and the salinity. Those are hopefully good because we're pulling that water in out of Resurrection Bay. So if something was off in that could be an issue. But we always have to make sure there's enough oxygen dissolved in there for the fish, but not too much. They can actually have too much gas dissolved in the water. So that's one of the tests that would be kind of like the equivalent weekly test. But I actually think they do that. I mean, they do that almost daily, I think, is go around and check the dissolved oxygen.
See, the more you know. I was learning about mammals and birds, and so I didn't get to ask really about the
I had
aquariums.
never seen this. So you got to tag along, and I had heard like, oh yeah, it's this 2-day process, and I'd seen them collect the little vials before, or I'd seen them walking around with those vials. But in my head, you know, and I think the public too, and we're like, oh yeah, we test the water quality. Maybe it was like a little pH strip that they dipped in there, or maybe they did add something to change the color overnight because I knew it was 2 days. I had no idea they were putting them in these little well packages and then basically like laminating those and sealing them up. So just watching you edit this video this week has been fun because every time I look over, I'm like, what are they doing? It's entirely new. So it was fun to see this process for the first time myself as well. It looks like maybe we've got another question. Oh, I'm getting the question tablet here from Alex. I love it. Oh, we've got some good ones here. Are there any animals that can be added to the pool or tank to help control bacterial overgrowth? So actually, we do have some animals here that help a bit, and they're not, unfortunately, in with the mammals or the birds. So like, for example, your scavengers and that sort of thing, they can clean up some of that waste. So things like sea cucumbers, right? We call them the sort of the Roombas of the SeaLife Center because they cruise around on the bottom. They suck up all the gunk down there, including waste from other animals, and they eat that. And you can't see it where our camera is, unfortunately. I probably won't pan us over. There have actually been studies at this, and one right next to us is looking at bioindicators. So this is not looking at control, but this is looking at other little organisms in the water that could indicate the overall health of that water. So that's something we've looked at or had studies done. But yeah, kind of just controlling the amount of waste. And unfortunately, if we put sea cucumbers in with our seals, they're just going to think they're toys. So they wouldn't last in there, unfortunately. But that's a great question. It looks like we've got another one here. What if you can't tell if it lights up? Because when they were shining the light, it fluoresced, right? Is it always that bright?
Yeah, so good question, and thank you for asking. Yes, so if it is going to fluoresce, it's going to be that like bright, like illuminating color. It's like a bright bluey purple color. So okay. Yes. However, whenever you're looking at the wells just with the naked eye, you saw the yellow, like the dark yellow, but some of them were not as prominent.
Oh, it won't necessarily get as orange.
No, some of them were not that like bright color. There were a couple where our team had to pull out like a sample well basically, or like a comparator well to use it as a reference point. And so they were able to kind of match them together and understand that the ones that got X's or slashes were just a little bit darker than what they were. So I have very much respect for that because I did not see a difference. No. But clearly there was a little bit of a difference.
I didn't notice a difference in it, but that does make me think more akin to like when I was growing up. We had like a fish tank in my house, and we had to check the pH. And yeah, you've got the strip, and then there's like the little compare strip, right? That you're like, oh, what color is it? So yeah, they don't all get that color. It's a great question. We got any more questions from you? All right, over there, we've got some here. Looks like how long is water quality take? So we mentioned before that it's about 2 days, but you said you had to incubate. Do you know kind of like how long are they doing this for?
Yeah, good question. So the first water bath for the Ent or the enterococci test was from about 18 to 22 hours of incubation, whereas the other one was around 24 hours. So once you saw Ryan putting them into the water bath, and then it kind of cut to the next day, whereas you didn't get to see the incubation. It's not too exciting to watch something just sitting in a little incubator.
Yeah, and you mentioned there's different types of incubators.
Yes, definitely. So you saw the water bath where it was, I believe, 41 degrees Celsius. So you put them upside down, so the wells that you saw were on the bottom, and then she put a weight on top of it to make sure they weren't going to float up to the top and they remain submerged. And then there's the other type, which is actually, you can kind of see it behind me, this red one with the red on it.
Oh, yeah, right.
Yeah, it was about 35 degrees Celsius.
And that was the one that was just in air. That's not what
right. Yeah. It has like this little slidey trays, and Ryan was slipping them in there.
Well, we have this question here in the chat, actually. Why are the wells yellow and not purple or green? And I would probably say it's the indicator that we use. That little vial, the little sachet that they crack open and dump in there, that's just how it reacts as it turns yellow. There likely are sachets out there that would turn different colors, but for the tests we're running, it's that yellow. And I know you've mentioned it, I've heard it from other people, the sachet for the enterococci, it kind of smells like farts or something.
Yeah, it has kind of like a sulfurous smell for sure. Yeah, it's definitely different. Yeah, the reagent packets, Natalie was talking about it in that last clip. whereas there's one that's specific for ENT and then one that's specific for total coliform.
Another comment
question.
here. If you find a water quality issue that does not pass, right, and it does happen from time to time, and especially like depending on seasons, if more stuff's going on in the water or less stuff, do you worry about releasing that water back into Res Bay? Ooh, this is a good one. I can answer this one.
This is a great one. Yeah, go for it.
And maybe the water you take from the bay, should that not be released back in? So yes, so for anyone unaware, we draw water in from Resurrection Bay. And now we also have filters, and this is going to have to be its own virtual visit at some point. We have a whole pump room downstairs with filters and all the pumps and sterilizing equipment. So for some tanks, we have sterilized them with ozone in the past, or other tanks will use ultraviolet light. So the water that we're pulling in from Res Bay, we clean that, and then we put it out into the tanks. And so if it gets really dirty in our tanks, we wouldn't want to necessarily dump like just really bacterial water back out there. So we actually will filter and sterilize it while it's in the facility as well. So that's a great question. But yeah, if something doesn't pass, we do have to take measures on our own stuff, because of course we want that tank to be cleaner. And it's not like the level that we're having as the pass-fail. It's not like that is the hard limit. It's just that's where the permit is, right? The animals are safe beyond that, but this kind of gives us an idea of like, nope, it's starting to get a little high. And so what do they do to sort of control that?
Yeah, so there are different measures that they can take. So they can either drop the pool halfway and clean it. They can scrub the sides. There's a whole list of different corrective actions, whether if it's in a certain range that they can be like, okay, well, this range, XYZ, results in this cleaning procedure.
Yes,
to make sure that our animals are totally safe. And then same with divers, like we would never send a diver in if there is some sort of issue. So it's always great to have this as like a checks and balances system.
Yeah, obviously, as the divers, humans, there's bacteria that the animals are going to be all right with, and humans aren't, and vice versa. So obviously, if some of those bacteria get real high, we don't want to have divers in there with like their face in contact with the water. So yeah, the test is done both for our animals, but also there's different limits for our divers. And you mentioned draining it, and for anyone that saw, gosh, we had an episode, I think it was actually our very first episode about
so long
diving
ago.
And cleaning the tanks, and around that time we actually did drain out all of SeaLife. Now that wasn't drained out to deal with a failed test or anything, it was actually drained to buff the windows. But if you watched that, it was a huge process to drain that tank. So some habitats here are not easy to drain, and we can't just drain them every day or whatever. So that's why we always want to keep a look or keep an eye on any test results, and we can increase water flow, we can increase filtration. I actually just saw another chat pop in there. Do divers ever get sick from bacteria in a natural body of water? It could happen. Now here, the danger would be if we just let that level go up. But that's why every Wednesday we're running this test. And in fact, I'm on the dive team. And so one of the emails I get every week is, hey, here are the results from water quality. So I've known the results for water quality every week. I just didn't know the process until
you didn't know how
Taylor took us back
intensive
here.
it was.
Yeah. So yeah, the divers could get sick, but that's why we test, is that if it's too high where they could get sick, then we say no divers today. We're going to take those corrective measures and either up the filtration or up the flow, or drain and scrub if we need to on some of those tanks, to make sure that it is safe for not only ourselves but the animals as well.
Absolutely.
Looking for other questions. Don't have any over there. The chat's looking pretty good. So I think we're probably good.
I think so too.
Yeah, I'm glad we've been able to share this. Just like selfishly, I'm glad, because again, this is something I've never seen it before. So it was exciting for us to be able to have Taylor come and do this, because again, it takes place on Wednesdays when we're doing our virtual visits. And these virtual visits are every Wednesday at 11 a.m. Alaska time. And as always, before we let you go, we want to thank our sponsors. So Royal Caribbean Group has made these virtual visits possible, and makes it so we can just air them out there free. And anyone that wants to kind of come to the SeaLife Center virtually is welcome to join us every Wednesday. And so with that, I want to thank you, Taylor, for taking us kind of back behind the scenes.
It was so fun. Thank you.
And I want to thank the audience for tuning in. And I hope we'll see you next week for another virtual visit from the SeaLife Center. So long.